Chromatography. Chromatography is a laboratory technique for the separation of a mixture. The mixture is dissolved in a fluid called mobile phase, which carries it through a structure holding another material called stationary phase. The various constituent of the mixture travels at the different rate, causing them to separate. The separation is based on the difference in the partition coefficient of stationary and mobile phase. Paper chromatography. Paper chromatography is defined as a technique in which the analysis of unknown substance is carried out mainly by the flow of solvent on specially designed filter paper. Types of paper chromatography. There are two types of paper chromatography. 1. Paper absorption chromatography. Paper coated with silica and alumina act as absorbent, stationary phase, and solvent as mobile phase. 2. Paper partition chromatography. Moisture and water present in the pores of cellulose fibers present in filter paper act as stationary phase and another mobile phase is used as a solvent. Principle of separation. The principle of separation is mainly partition rather than adsorption. Cellulose layer in filter paper contain moisture which act as stationary phase and organic solvent/buffers are used as mobile phase. Instrumentation. Stationary phase and paper used. Application of sample. Mobile phase. Development technique. Detection or visualizing agent. Stationary phase and paper used. Wattman filter papers of different grades like number 1, number 2, number 3, number 4, number 20, number 40, number 42 etc. are used. In general, this paper contains 98 to 99% of alpha cellulose, 0.3 to 1% beta cellulose. Factors that governs the choice of paper. Nature of sample and solvents used. Based on quantitative and qualitative analysis. Based on the thickness of paper. Types of papers used in paper chromatography. Modified papers, acid or base washed filter paper. Hydrophilic papers, papers modified with methanol, glycol, glycerol etc. Hydrophobic papers, acetylation of OH group leads to hydrophobic nature, hence can be used for reversed phase chromatography. Coating of silica, alumina can also be made. Preparation of paper. Cut the paper into desired shape and size. The starting line is marked on the paper with pencil 5 cm from the bottom edge. On the starting line, marks are made 2 cm apart from each other. Preparation of the solution. Choice of suitable solvent for making solution is very important. Pure solution can also be applied direct on the paper, but solids are always dissolved in small quantity of suitable solvent. Application of sample. The sample to be applied is dissolved in mobile phase and applied as a small spot on the origin line using capillary tube or micropipette. Very low concentration is used to avoid larger zone. The spot is dried on the filter paper and placed in developing chamber. Choice of solvent. The commonly employed solvents are the polar solvent, but the choice depend on the nature of the substance to be separated. Mobile phase. Pure solvents, buffer solutions, or mixture of solvent. Example, hydrophilic mobile phase. Isopropanol, ammonia, water 9:1:2. Methanol, water 4:1. Hydrophobic mobile phase. Dimethyl ether, cyclohexane. Kerosene, isopropanol. Chromatographic chamber. This is made up of many materials like glass, plastic, stainless steel. Glass tanks are preferred most. They are available in various sizes depending on paper length and development type. The chamber atmosphere should be saturated with solvent vapor. Development technique. 1. Descending type. Development of the chromatogram is done by allowing the solvent to travel down the paper. Here, mobile phase is placed in solvent holder at the top. The spot is kept at the top and solvent flows down the paper from above. 2. Ascending type. Here the solvent travels up the chromatographic paper. Both descending and ascending paper chromatography are used for the separation of organic and inorganic substances. The sample and solvent move upward. 3. Ascending-descending type. This is the hybrid of both of the above techniques. The upper part of ascending chromatography can be folded over a rod in order to allow the paper to build descending after crossing the rod. For circular chromatography type, radial. A circular filter paper is taken and the sample is deposited at the center of the paper. After drying the spot, the filter paper is tied horizontally on a petri dish containing solvent, so that the wick of the paper is dipped in the solvent. The solvent rises through the wick and the components are separated into concentric rings. 5. Two-dimensional type. In this technique a square or rectangular paper is used. Here the sample is applied to one of the corners and development is performed at a right angle to the direction of the first run. Detecting/visualizing agent. If the substance are colored they are visually detected easily. For colorless substance physical and chemical methods are used. Physical method. Example iodine chamber and UV chamber method. Chemical method. Example ferric chloride for phenolic compound and ninhydrin in acetone for amino acids. RF value, retardation factor. In paper chromatography the result is represented by RF value which represent the movement of solute relative to the solvent. Factors affecting RF value. The temperature. The purity of solvent used. The quality of paper, adsorbents and impurities present in adsorbents. Chamber saturation techniques, method of drying and development. The distance traveled by the solute and solvent. pH of the solution. Applications. Separation of mixture of drug. Separation of carbohydrates, vitamins, proteins etc. Identification of drug. Identification of impurities. Analysis of metabolites of drug in blood or urine.